everybody. Welcome to the Biological Chemistry series of Bio 181. Now that you have a basic understanding of atoms and the chemical foundation of life, we are going to discuss the major organic molecules that make up living organisms. In this series of lectures, you will come to understand the structure and function of carbohydrates, lipids, proteins, and nucleic acids. I always like to review where we've been and where we're going. So far, we've learned that the building blocks of all matter, atoms, which bond to form molecules. And in this lecture, we're going to be discussing how these smaller molecules form complex interactions to form macromolecules. Once we understand these macromolecules, we'll be able to learn how these interact to form the fabric of living organisms, which is membranes and other biological structures like organelles. First, we need to understand some basic terminology. Macro means large, and the large molecules we are talking about are made of smaller molecules that are bonded together. Often these are repeating subunits, but we will discuss these subunits for each macromolecule this coming week. However, a basic term for these subunits is monomer. A monomer is a small subunit of a macromolecule. Typically, monomers are the smallest unit of a certain macromolecule. For example, carbohydrate monomers are composed of rings of carbons. Glucose, the carbohydrate that is measured for blood sugar, is a single sugar ring. In other words, a monosaccharide. If that sugar ring is broken down, the resulting chemical would no longer be considered a carbohydrate. In other words, monomers are the smallest subunit of a given macromolecule. Many macromolecules require bonding between numerous monomers, a process called polymerization. When a large macromolecule is composed of many subunits bonded together, it's called a polymer. For example, a string of glucose monomers can polymerize or bond together to form a polymer called glycogen. So how do monomers become polymers? As mentioned, via a process called polymerization. But how does polymerization take place? Polymerization reactions are examples of dehydration reactions. As you can infer from the name, dehydration reactions involve the removal of a molecule of water. This involves a hydrogen from one monomer and a hydroxyl, or an oxygen and a hydrogen group, from another monomer being removed. So two hydrogens and an oxygen, which leaves the ends of these monomers open for bonding. As you can see an example of a dehydration reaction taking place in this image. You see the water leaving here. As with most reactions, dehydration reactions are facilitated by enzymes. So we remove a water molecule to create a bond between monomers, and therefore logically, we add a water molecule to break a bond between monomers. For example, breaking polymers into monomers. A reaction that uses water to break a bond is called a hydrolysis reaction. The prefix hydro refers to water, and the suffix lysis means to break. Thus, these reactions utilize water to break bonds. Okay. So now we know the basic terminology for macromolecules, as well as the major reactions that assemble or disassemble them. Now let's discuss their structure. All biological macromolecules contain carbon and hydrogen. Usually these atoms compose the structural backbone of these macromolecules. Collectively, molecules that contain a backbone of carbon and hydrogen are called hydrocarbons. These are the foundation of organic molecules. Many organic molecules also contain oxygen and nitrogen, but not all. All do, however, contain carbon and hydrogen. Carbon turns out to be an excellent atom to form the backbone of molecules with strong structural integrity. This is because of its instability. Okay, that sounds really funny. But remember from our atoms and bonding lecture that the more unstable an atom, the more likely it is to form bonds. Carbon specifically has 4 valence electrons, meaning that it very easily can share electrons with 4 hydrogens or 2 oxygens or 1 oxygen and 2 hydrogens, etc. This means that it is happiest when sharing electrons and readily forms covalent bonds, which, if you recall, are the strongest types of bonds. Thus, carbon-containing compounds tend to lend structural integrity to these important biological macromolecules. Aside from a hydrocarbon backbone, macromolecules also tend to contain a variety of what are called functional groups. These are groups of atoms that are commonly found in macromolecules and exhibit certain functions. These include hydroxyl groups, which we've discussed briefly, saying OH, but also carbonyl, carboxyl, amino, phosphate, and methyl groups. These are not the only functional groups that exist, but these are the common ones found in biomolecules. So what do these functional groups do? What is the function of these functional groups? Well, amino groups are weakly basic, and as mentioned by this chart, are really good at accepting hydrogens. As we will see, these are characteristic of amino acids, which are the monomers of proteins. Carbonyl groups, which are found in lipids like steroids and waxes, as well as proteins, are excellent at forming hydrogen bonds. Carboxyl groups are acidic and are found in amino acids and fatty acids like proteins and lipids. Hydroxyl groups, which are abundant, are found in most macromolecules and are excellent at forming hydrogen bonds with water and are often a functional group involved in hydrolysis or dehydration synthesis reactions. Methyl groups are nonpolar, thus they are repelled by water. These are present in DNA and proteins and some carbohydrates as well. Phosphate groups are characteristically present in nucleic acids, but are also present in other macromolecules. These are polar and weakly acidic. Along with hydroxyl groups, these groups interact to form the backbone of DNA and RNA. Sulfate groups contain sulfur and are a common component in proteins and lipids, and sulfhydryl groups are characteristic of the amino acid cysteine in particular. While you do not need to memorize these groups or this table, it is important that you are able to recognize these functional groups in the macromolecules we discuss. This table summarizes the 4 macromolecules that we are going to discuss in the remainder of this lecture series. We will discuss each of these in detail: carbohydrates, lipids, proteins, and nucleic acids. These are the terms and concepts that you should focus on in this lecture. This lecture in particular just served to introduce you to the macromolecules as a whole, but moving forward, we're going to discuss each group a little more specifically. For our upcoming lectures, please plan on having these listed items available, if you are able, for some small experiments that will take place in the middle of those lectures. Please, for the carbohydrate lecture, it would be nice if you had a saltine cracker. And for the lipids lecture, please have potato chips or maybe like butter or olive oil, and either a brown paper bag or a piece of construction paper. Thank you again for being a part of our class. Jamie and I are so happy you're here.